So thanks, John, for the invitation and for organizing the workshop. I'm excited to see all the cool research that will be shown in the workshop, and I'll present any updates on the research that the group I'm in is doing about building a atomic level resolution model of the JCVI Sin3A. So in the group in Groningen that I'm part of, we mainly work on molecular dynamics simulations, which we use as a tool to study a large variety of biomolecules. The classical way to do these molecular dynamics simulations is by taking an atomistic structure of a protein or a small molecule, then calculating the Newtonian equations of the Newtonian forces, and then integrating them and getting the dynamics of these models. So you can do this for a large set of biomolecules. But what our group focuses on is not these atomistic simulations, but mainly coarse-grained models. So we don't represent every atom in every biomolecule, but we group together multiple atoms into pseudo-atoms, or what we would call beads, to reduce the complexity of these models, which allows us to simulate larger structures, but also for longer timescales. There's no free lunch in these computational methods, so you lose a bit of detail while you do this coarse graining, but it has been shown to be a very useful tool for a large set of applications. So the particular flavor of coarse grained model that we do is called the Martini coarse grained model. So it's a type of coarse graining where we have a systematic set of rules that you can apply to any biomolecule to then create a coarse grained model of a large set, almost all biomolecules, going from proteins, lipids, nucleic acids, and metabolites. So since we can create a coarse grained model of all these different biomolecules, the next logical step would be to think about whether we can combine them and create a model of an entire cell. So that's the project I'll talk about, and I'll go through the individual steps that we take to eventually end up with a coarse grained model. So we didn't have to start from scratch. So John already showed this picture. We started from the molecular of the simulations that the group of Sen Schulten that they have performed at the UIUC, where they built a whole cell kinetic model. So it's not a model that focuses on the molecular motions, which is a type of simulations that we do in our group, but it's a type of simulation that looks at metabolic fluxes. So they look at which reactions happen where in the cell, and they try to make some predictive statements about what's the cell doubling time, which they have been quite successful at. So it has been quite recently published in Cell. And to make these models, they had to gather a lot of information, so a lot of proteomics and metabolomics information, and etc., also lipidomics, which they use to create this very accurate model. So what we do, so we work in quite close collaboration with the group of Zen, and then we take all the information that they have gathered around the cell, and we take them as an input to create our own molecular models. For the first version of the model we're building, we decided to work at a practical scale. So building at the full size of the cell is quite large for classical MD simulations, since cell sizes are quite large for us. So what we decided to do is we decided to shrink the cell by a factor of 2, reducing the radius of the cell from 200 to about 90 nanometers, which would result in a decrease in particles by a factor of 10. So the aim we have for this first version of the model is that it's smaller in size, but that the complexity would be as high as possible. So we would try to include as many different constituents, molecules as possible, so that we can create a quite complex simulation, but at a feasible size for now. So the start of this whole cell modeling process is a chromosome. So this is again in collaboration with the group of Zen at the UIUC, where they created a very mesoscopic coarse-grained model of the chromosome, where they represent 10 base pairs by a single bead. This is quite a coarse resolution, but they were able to quite nicely capture the fractal globule structure of this chromosome, this very specific structure, and they were also able to capture the change of the chromosome during the cell division cycle. So basing our model on this chromosome structure, we can build a cell structure during various stages of the cell division cycle, in theory. The only thing we have to do is we have to translate this type of model into the Martini ecosystem. So we have to map it to a Martini resolution, which is significantly higher or more detailed than what is shown here on the screen. So we devised a protocol to do this. So we interpolated all these coarse beads and scaled them up to approximately a near atomistic resolution, where we captured a nice helical structure of the DNA. I won't go into details on how we performed this mapping, But to verify our mapping, we ran a few hundred nanoseconds of simulation of a chromosome of the Century A, which is shown here on the screen. I hope the movies translate okay over the video call. But to validate our model, we ran the simulation and we looked at how fast these individual base pairs diffused and also how bendy the chromosome is. So we calculated a quantity called the persistence length to quantify this bendiness. And the values we got out was quite close to what you would expect from a real chromosome. So this validated our process. Having modeled the chromosome, the next step would be to model the cytoplasm which goes around the chromosome, which we did by first starting to look at the proteome. So using the genome and the annotations in the genome, we can quite accurately, using structure prediction methods like AlphaFold, go from the genome to a chromosome up to a protein structure. However, some of these proteins don't occur in monomers, which is something that AlphaFold mainly focuses on. But especially for the membrane proteins, it's quite important to also capture the oligomeric state, so the structure of the complexes of these proteins that are formed. So we also decide to map the proteome and see if we can predict these multimeric structures. We did this by looking at sequence similarity, at interaction partners, by looking at the string database, which is a quite widely used database to see which proteins interact with other proteins based on a wide range of experimental measurements. And we also looked at structural homolog. So we combined AlphaFold with FoldSeq to map the possible structurally similar multimers or monomers to what's already known in literature. Doing this, we got a quite diverse set of protein structures, which range from proteins that largely occur as monomers to quite large complexes, mainly shown here on the screen. Overall, they were quite highly confident structures, and these can then be used to incorporate into our cellular model. A nice side quest or a side tangent that came out of this project is that also these new structures, these new multimeric structures, can be used to predict possible uncharacterized genes. So if we predict multimeric structures of these uncharacterized genes using structural homology searches, we can then map possible functions of these genes onto them. And for about 30% of the uncharacterized genes, this gives us a quite decent approximation of, or a quite decent guess of what these uncharacterized genes could be doing. But in our model, we mainly use these larger complexes to incorporate them in the cell envelope, since that's the region where these protein complexes for us are the most important. So we gathered all the proteins, structures and protein counts based on proteomics data. We gathered the RNA, which we did by translating the chromosome, and a large suite of metabolites. And we packed them into the cell, which we now have as a quite high throughput approach. So we can make these cell models in a few minutes, which opens the door of maybe making or incorporating our cell modeling pipeline in a high throughput approach in the relatively near future. The last element of the cell that is still missing is the membrane. So we have the chromosome with the cytoplasm around it. We now still have to encapsulate it with a membrane, which we base on the work that the Science Lab has been doing at TU Dresden, where they looked at these very minimal lipidomic sets. So they looked at only cholesterol and POPC and cholesterol and DOPC, or diether PC, sorry. And we used these very simple lipid compositions to reduce a bit the complexity of the cell membrane. However, we did capture the protein variety in the cell membrane quite accurately. So we took all the protein models we created previously, we inserted them into small lipid membranes to get their orientation. We did this for the whole set of membrane proteins, which may be quite small on your screen, but there's quite a diverse set of membrane proteins. And we simulated them for a few hundred nanoseconds to verify the stability, and then later inserted them into a spherical cell membrane, which can then be put around the cytosol to create a full cell model. And the one thing that is quite striking here in the model that we created is that if you calculate the protein area of the cell membrane, visually it already looks quite high, but if you calculate it, it's only about 25%. So the number of proteins we inserted was based on proteomics measurements. However, the occupied protein area is quite a bit lower than what you would expect from the literature value. So you would expect about 50% of the cell to be covered by proteins, but in this case, it's only 25%. So this might indicate an artifact or a proteomics artifact in the trypsin digestion that is used to calculate the proteomics on which we base our cell modeling pipeline. But this is something we still have to further investigate, but it's a quite nice illustration of how making these simulation models, these computational models, also can give some indication or can give some insight in experimental measurements, which I think was quite an interesting result and definitely worth further looking into. So I think we're in the current data set we have for the proteomics, we're somewhat underestimating the number of membrane proteins that we insert or that we count in the cell. Yes, so finally we built a cell model. The only thing that rests us to do is now simulate it. There's quite a few things that can go wrong. So over the last year, we've been struggling a bit with getting this cell to simulate. So on the screen, you see a tornado cell that we by accident simulated. This very large deformation in the cell was only caused by a tiny parameter in the cholesterol that we use in our simulations, which was not correctly balanced, but that leads to these very large global deformations, which shows you that we have to very consciously curate all the ingredients we put inside the cell, illustrating also how difficult or how quite laborsome this large-scale modeling effort is. However, we're getting there. We're making decent progress, so these issues have been solved. So if we then go through the steps again, so we start with the chromosome, we built a membrane around it, we inserted all the proteins and the RNA, we sprinkled in then some metabolites, and then we run the simulation. So you won't see that much movement since it's only a simulation of 100 nanoseconds, which is the start of what we're now trying to do. But using these dynamic informations, we already can start to think about analyzing protein-lipid interactions, maybe protein-protein interactions, and really getting some scientific data out of these computational models. The one thing that you might see on the screen is that there's also some holes forming in the membrane. And these holes indicate that we didn't accurately enough calibrate the lipid concentration we added in the membrane. So there's still some tweaking that we have to do before we reach our final cell model. But we're getting quite close to connecting, hopefully, our computational models somewhat directly to also experimental measurements, which is something I'm excited to think about during this workshop, whether we can maybe use our computational microscope, which we like to call it, or simulations to connect to experimental measurements. So yeah, that's the conclusions is that we're very, very strong on the way to build these cell simulations. We solve most of the problems, and the perspectives we have is that we can use these simulations we run to maybe inform certain experimental measurements. We can improve our understanding of the JSVI Syn3A, and we can start analyzing these in-cell dynamics and see how the dynamics of the inside of the cell really behaves like, which I think is quite exciting, and I'm quite curious for the things we might find in our large-scale models. So I'd like to thank everybody that helped me work on this large simulation effort, and if there's any questions, I would like to answer them. Thank you.
Very nice, Jan. My first thought is you talked about the low number of proteins on the surface of the cell. So remember that the minimal cell and the Mycoplasma mycoides cell on which it is based are about the same size. And
yes,
we have gotten rid of 90% of the surface-associated protein encoding genes that are in the wild-type cell. So it would seem to me that the protein composition on the surface of the cell ought to be pretty sparse. But I don't know how I don't know how to measure this.
Yeah, so it's something that in theory we're quite extrapolating for the fact that we expect or we think that this concentration might be too low, but it seems to be quite a conserved quantity, that most of these cells have this very dense outer membrane. And the reason that we made this, or we're looking into whether or not the concentration is too low, is that also in the paper by the Lucy Shilton lab on these kinetic models, there they really see that in their models, if they make rough approximations, that the protein density they would expect would be 50%, but they do this on like a very rough approximation of what the size of these proteins is. So that's what we're comparing to, and that's where we saw this discrepancy. But I think it would be very interesting to see if there is some experimental maybe an AFM or a fluorescent measurement that we can do to see if the density of these proteins actually is much lower than what you would see in a regular, let's say, E. coli cell or mycoplasma.
Does anyone in the audience have a sense of how you might measure this? I don't know if this is something AFM can do or not.
John, this is James Dobbins Speck at UAB. First of all, I think he's missing a set of proteins that will be on the surface, and I think that 50% number sounds about right to me after growing the cells and working with the proteome of the membrane. The first set he's missing is the moonlighting proteins that are on the surface, and I'm in about 20 minutes or 30 minutes when I talk, I'll show the data to back that up. But there's a significant percentage of enolase, DNAK, EFTU, all these moonlighting proteins that are classified as cytoplasmic enzymes. That's enough to account for that extra 25% that he's missing.
So this is something we haven't actually worked on yet, but I think this is one of the ideas you also had. Like, you're using our computational approaches, we can try to screen whether or not we are missing some of the transmembrane proteins that we would classify as cytosolic proteins by just recognizing whether or not we can embed them in a membrane and whether they would stably be embedded. So if you have a set of proteins that could be used for this, that would be very interesting.
These proteins are clearly on the surface of the Synth3A cell. Enolase, DNAK, and EFTU, I know for sure. I have the data to back that up. I'll show it here in a minute. Like I said, there is a significant fraction, and these are high abundance proteins. That's why John Sanford, who's going to talk here in a little bit, and I both find them, because they're very high abundance proteins on the surface. Enolase, EFTU, DNAK, there's a whole bunch of them.
But that's still
that will make up that extra 25%.
But guys, we've still got, think about that with SYN1. Again, I don't have quantitative proteomics for SYN1 or wild type Mycoplasma mycoides, but we've gotten rid of 90% of the genes that have transmembrane regions.
True. Very true,
and
John.
So,
but those are very low population proteins.
Are they?
Yes. On the mycoides surface, there might be 50 copies, where there's 1,000 copies of enolase.
Okay.
But I think the literary value of 50% is usually measured by transmembrane helices. So I think a DNAK would like
these cytoplasmic proteins are not attached by transmembrane helices. Period.
Yeah, so that's why I'm curious to see whether
in here in a minute. But I guarantee you they're not. They're attached through a glycophospholipid linker.
Okay. So I've got a collaborator at MIT who's going to redo the quantitative proteomics, both for SYN1 and for minimal or for wild type Mycoplasma mycoides and minimal cell, maybe with closer to the state of the art proteomics than what we have. The data that we're using now was generated 6 years ago. And so we'll see what we get. That'll be something that hopefully we'll either get published or certainly talk about next fall. More questions for Yen.
Well, perhaps I could add something to Jan. I remember old work from Don Engelman, who has actually determined, let's say, the density of integral membrane proteins in a number of membrane types. And then the membrane is actually way more crowded than what we are seeing in this image. So I would say, Jan, what you see is a rather dilute membrane in terms of membrane proteins, at least what I can see from the outer surface. And I think that might be in line with what John is saying, that, well, they have removed, 80 or 90% of the integral membrane protein. So to me, this looks pretty okay if you compare it with old proteomic studies of Don Engelman's lab in Yale.
Okay, that's interesting. Yeah, so the reason that this question came up also is that I think for trypsin digestion, which is a method I think was used to get these proteomics measurements, is that the membrane proteins usually cause or can cause or are known to cause some artifacts, but it could be very much so that this is just how what you would get for a JCVI Syn3A. But I think comparing it to maybe other models that have been made about other cells, I think the mycoplasma genitalium by the Goodsell lab, I think that's something we then can compare to see if our methods overlap, let's say.
And genitalium is going to be a hard one because it's not a sphere. So
yes,
it's going to have, as you spread the thing, as you string the thing out, the surface area to volume is going to go way up. The other thing that maybe the Sayans group can say is they may be able to calculate what is the total area of the lipid, the membrane of the surface, as opposed to the proteins. Sorry.
Yeah. So that would be interesting to look at, definitely.
So Jan,
James, are you on?
You also mentioned that you had too few lipids, and as a result of that, you had a hole in the membrane.
Mm-hmm.
Why doesn't your cell shrink? Why doesn't it adjust?
So I think the reason is that we based our number of lipids to insert into the membrane by looking at the area per lipid of a flat membrane, which is an approximation that we made. And then if you start simulating them, I think getting a large deformation in the cell is too costly in energy. Is a too high energy penalty. And then what happens is that at some point the water will just push out and create a pore, and then the pore will open and never close again. I think that's the mechanism.
Okay, thanks.